Fasting is widely celebrated for its ability to trigger autophagy, a cellular self-cleaning process that removes damaged components, supports mitochondrial renewal, and preserves tissue function as we age. But as intermittent fasting and other time-restricted eating strategies become more popular, a key question emerges. Can multivitamin supplementation enhance the benefits of fasting-induced autophagy, or might it disrupt the delicate nutrient-sensing balance that makes fasting effective in the first place? The answer lies in understanding how specific micronutrients interact with the cellular stress signals that activate autophagy, and whether supplementation during fasting provides critical support or counterproductive interference. At the heart of autophagy is a tightly regulated nutrient-sensing network. When nutrient availability drops, especially glucose and amino acids, cells interpret this as a signal to initiate conservation and repair. Energy-sensing enzymes like AMPK are activated, and growth-promoting signals like mTOR are suppressed. This metabolic switch encourages the cell to break down non-essential or damaged components to recycle resources. Fasting is a powerful natural trigger of this process. However, autophagy is not just about withholding nutrients. It's also about managing oxidative stress, maintaining mitochondrial health, and preserving enzymatic function during the repair phase. These supportive roles are where multivitamins may play a beneficial part. During fasting, certain micronutrients become especially important because they act as cofactors for enzymes involved in the autophagic machinery. B vitamins such as B2, riboflavin, B3, niacin, and B6, pyridoxine, are essential for energy production in mitochondria and for the conversion of fatty acids and amino acids, key fuel sources during a fasted state. Without sufficient levels of these vitamins, the body may struggle to sustain energy balance while cells are engaging in autophagic activity. Moreover, niacin serves as a precursor to NAD, a molecule required for sirtuin activity and for driving longevity-associated transcription factors like FOXO that promote autophagy and mitochondrial repair. In this context, low-dose supplementation during fasting may not inhibit autophagy, but rather enhance its efficiency by providing the necessary metabolic tools. Antioxidant vitamins, such as vitamin C and E, also have a complex relationship with autophagy. While oxidative stress is a natural trigger for autophagy, uncontrolled or excessive oxidative stress, especially during prolonged fasting, can damage lysosomal membranes and autophagosomal structures. A modest intake of antioxidant vitamins may help buffer this stress, protecting cells from collateral damage while allowing autophagy to proceed. Vitamin C, for instance, supports iron regulation, which is crucial for lysosomal function, while vitamin E stabilizes cellular membranes under metabolic strain. In short-term fasts, the body often manages without external antioxidants. But for longer fasts or in individuals with marginal micronutrient reserves, multivitamin support could potentially prevent cellular injury and optimize recovery. Vitamin D and K2 add another layer to this interaction. Both have roles in inflammation modulation, mitochondrial function, and cellular gene expression. Vitamin D, in particular, has been shown to support autophagy-related gene transcription via its interaction with the vitamin D receptor, VDR, which modulates dozens of genes related to immune resilience and cellular turnover. Supplementation with D and K2 during fasting periods, especially in individuals with pre-existing deficiencies, may strengthen the long-term regenerative effects of fasting, helping the immune system, bones, and vascular system benefit more fully from cellular renewal processes. The timing and formulation of the multivitamin matter greatly. High doses of certain nutrients, especially amino acids or iron, may activate mTOR or other growth-related pathways, potentially blunting the autophagy response. However, carefully calibrated multivitamin blends that avoid excessive macronutrient triggers can be designed to support enzymatic functions without disrupting fasting signals. This is especially true for low-calorie or zero-calorie vitamin formulations that use bioavailable forms of micronutrients without sweeteners, fillers, or added calories. From a systems perspective, autophagy and nutrient availability exist in a feedback loop. While fasting initiates the process through deprivation, optimal execution and resolution of autophagy may depend on the availability of key micronutrients for energy conversion, detoxification, and tissue repair. Individuals with micronutrient deficiencies, especially common in older adults or those on restrictive diets, may find that fasting depletes them further, reducing the efficiency or safety of autophagy. Strategic supplementation in these cases could help ensure that the benefits of fasting are fully realized without unintended stress. In practice, this suggests a potential middle ground. Taking a well-formulated multivitamin with water during a fasting window may not only be safe, but also beneficial for many people, particularly if it avoids nutrients known to raise insulin or mTOR activity. Water-soluble vitamins like the B-complex and vitamin C are more likely to support fasted biochemistry, while fat-soluble vitamins like D and K2 may be best absorbed with small amounts of dietary fat, often consumed at the breaking of a fast. For longer fasts, 24 to 72 hours, small supplemental doses may help maintain metabolic integrity and prevent depletion. Ultimately, the effectiveness of multivitamin supplementation during fasting depends on individual context, baseline nutrient status, length and intensity of the fasting regimen, metabolic health, and fasting goals, e.g. autophagy versus fat loss versus immune reset. For those with nutrient-rich diets and short fasts, additional supplementation may not be necessary. But for others, especially those pursuing anti-aging or regenerative fasting protocols, a targeted, fasting-compatible multivitamin may serve not as a crutch, but as an amplifier of the body's intrinsic repair systems. In sum, rather than disrupting fasting-induced autophagy, the right micronutrient support may reinforce it, helping the body recycle, rebuild, and rejuvenate more efficiently. As we shift toward precision health approaches, integrating fasting with intelligent, personalized supplementation may offer the best of both worlds, the cellular benefits of stress and the structural support of nourishment.